Hello everyone. This is Neslihan from COGIS, which is a community where students and professionals from various fields of study gather around to make resources based on cognitive sciences more accessible and provide students a way of communication with the various events we hold throughout the year, such as academic cognition, which is a series of talks preferably given by undergrads and master's students. Book cognition that provides an opportunity for professors and students to exchange ideas revolved around a book of choice for several weeks, and Growing Up in Science, where professors share their lifelong experiences about the academic life. You can follow our accounts on Instagram, LinkedIn, and Twitter, and you can also support us from the patreon.com, and we would truly appreciate your support. Today's event, named the Cognitive Webinar, is designed to be online and held entirely in English, aiming for an international audience. We would like to provide a platform not just for science communication, but also an interaction of academics from all over the world. Today, we're going to listen to Dr. Collignon, who is a cognitive neuroscientist at UC Leuven in Belgium, where he also got his PhD from. He completed his postdoc at the University of Montreal. He is currently the primary investigator of the CPP lab. His research is funded by various organizations, including the European Research Council, by focusing mainly on the mechanisms underlying cross-modal perception and plasticity in the brain, as well as how changes in multisensory experiences shape the architecture of the mind and the brain. Today, we're going to listen to his talk entitled, "What Does Blindness and Deafness Tell Us About Brain Development?" His talk will last about 40 to 50 minutes, and then we will give a 5-minute break. After the short break, we will proceed with the Q&A session, which will also last about 30 minutes. Before we proceed, I would like to emphasize on the fact that we would like our attendees to ask their questions with their microphones and video cameras open if possible, but it's not a requirement. You can also ask questions through the Zoom chat as well. And Dr. Collignon, please take us away whenever you're ready.
All right. I'll just start by sharing my screen. Do you see it?
Yes.
Perfect. So thank you very much for the invitation to present my work. I'm very happy to be here. I've been told that the audience might be a bit diverse somehow. So I don't know, I prepared a talk that is also somehow a mix between like a more general introduction to the topic, but also with some specific empirical details. But I'll not go in the details of the technique that I am using. So I'm very happy that there is this long session after the talk. Please feel free to ask me any questions after the talk if something is not clear. And yeah, let's directly jump in the topic. And I like to start with this painting that I like very much. Probably most of you recognize this painting. It's called The School of Athens by Rafaello. It's in the Sistine Chapel, right? And here, the most two prominent figures that we see at the center of these screens are an older Plato and a younger Aristotle, right? That apparently were already discussing these somehow very long-standing debates of how nature and nurture interact in shaping brain development, right? And there is something of interest here in this way. These two prominent scientific figures are represented. Plato is pointing his finger toward the sky, like somehow suggesting, at least Rafaello is trying to suggest, that he was defending a view where knowledge somehow comes from above, like somehow is inherited in the human brain, while Aristotle is pointing his hand to the ground, like if he was sensing it, and somehow suggesting that to build knowledge in your brain, you have to interact with the world and sense the world to create this knowledge in your brain. So this obviously created two very prominent field of philosophical debates, I would say, like the Nativism versus Empiricism, like people like let's say Descartes, a strong nativist person, and John Locke as a strong empiricist, people debating about whether nature versus nurture are the prominent features that shape the building of human knowledge in the human brain. Obviously, we know now that this is kind of a trivial dichotomy, right? Both these things interact in shaping brain development. And if we put it in a more modern perspective, we could ask ourselves how these two things, like how nature and nurture, interact in shaping the functional tuning of brain regions, right? So how brain regions adopt the sensory and cognitive tuning to the operation they are tuned to, right? So, and we know that nature is in the place. I'm just like receiving this message that I'm not supposed to let people enter, right? You will do that, I guess. I have this message popping out to ask me if I can let.
Yeah, we're handling that.
Okay. So the idea is that nature is in the place, right? So, but we know that somehow the brain is a product of a long evolution, right? So we know that the human brain is the byproduct of a long period of development, and if we go back into our let's say the trees that precedes us, what we see is that one very salient feature of brain organization is the segregation of the sensory systems, right? You have different regions in your brain that prefers to react to some specific sensory inputs, right? Here. And this is true even for our oldest ancestors or cousins on the evolutionary tree, right? And if we zoom in one of these systems, like for example, the prominent visual system in the brain, right? We see that inside each of these regions that prefer one sensory modality, there is also an organization, right? Typically a hierarchical organization where you have primary sensory regions that have very small receptive fields and tuned to some very specific low-level features of the visual environment, like for example, orientations, like in V1. And then while you progress in information processing in the ventral visual stream, for instance, your receptive field will enlarge, and these regions will start to be tuned more and more to more conceptual information, if you want, right? Is it something that is living versus non-living? Is it a face? Is it a tool, right? And this somehow independently of the low-level features of these objects, right? More the categorical or conceptual representation of these objects. And so why do I say like that nature is in the place is that, for example, if you take V1, the early visual cortex of a young kitten at P14, this is the moment the kitten opened the eyes, and you look at this tuning for low-level features like orientations, right? We know that in V1 there are columns that prefer some specific orientation. This is what these, let's say, this display is representing. This is a patch of V1 in cats, actually, and you see that there are zones in these patches that prefer some orientations, right? So, and this is probed by microelectrodes, and you see that there are these patches that appear, but even at P14, when the kitten is just opening the eyes, so basically it has no visual experience, there is already this patchy organization. This organization refines with time, right? But it's already there, right? It's already implemented in their brain. We also know that when we move on in the visual hierarchy, right, that there are like these tunings, like I said, toward like more high-level features of the visual environment, like places, faces, tools, words, or moving objects, right, that have a specific patch of the occipital cortex that prefer these specific stimuli. And even for these more complex stimuli, it seems that it's built in, right? So there are some studies, like this recent study here, that show that already 15 to 90 hours after birth, right? So basically these human babies have almost no visual experience, where you present them shapes like this that look like a face, have this triangular structure versus reversed shape, let's say reversed face, the brain, the occipital cortex of these babies react more to this face-like stimuli versus reversed face-like stimuli. Like if already 15 hours after birth, there is already some tuning to face-like stimuli, right? And actually, and again with time, like after 4 to 6 months old, you present babies to such a very complex visual scenes, like here, right? And you will see that the participant does not know, but sorry, at specific frequencies, there are like phases that reoccur in these stimuli. And what you see is that you have already 4 to 6 months old, a very sharp face selective response after that. So it's inborn and refined with time, and very quickly, 4 months after birth, you can present very complex stream of information like this one, and you will see that the brain tuned to faces compared to other objects. Right, so it's kind of a wonder, right? That actually, as quick as that, the brain seems to tune to very complex visual stimuli. So I've just demonstrated that nature is in the place, but obviously nurture is also in the place. The problem, though, is that it's very difficult, actually more difficult somehow to display. Why is that? Is because as humans, even if we think about ourselves as very idiosyncratic individuals, we, at least at the macroscopic level, have very redundant experiences. We all see faces, we all develop, most of us develop languages, we use tools, we interact with and navigate in our environment. So at this level of macroscopic brain organization, across cultures, actually, we have a very, very similar experience of how we interact with our environment. And somehow one way to disclose this effect of nurture is to go to the extreme. And so basically, the idea in my lab and in the lab of many other people around the world, the idea was to use people having extreme experience of how they interact with the world, like blind or deaf people, right? So people that miss one sense. And the idea here is to look in the brain regions that as I've just said, are tuned from the start to process visual information in an organized fashion. What do these regions become, right? If experience has no role, these regions, they probably will stay silent, right? But if experience can shape brain development, we should say something happening in these regions. So one first thing that people have done is just deprived, like I've said, the kitten, for example, from vision. And you see here kitten raised in the dark, for example, right? So what you see is that kitten raised in the dark, at P14, they have this kind of patchy organization. It's normal, right? It's inborn, like I said, it's independent from experience. But if you let grow the kitten in the dark, in darkness, they will lose this organization, right? At P45, so 45 days after birth, if you don't provide the kitten visual information, this patchy organization for vision disappears. So the structure that is predetermined is kind of moving, right, away from its a natural way of processing information. This has been also recently demonstrated in another study where they've been raising primates without these primates seeing faces. And what they see is that if you raise for 1 year these primates without seeing faces, they do not develop afterward a face selective patch, right? So basically you need such experience. It's there, but you need somehow some experiences, visual experiences early in life to to maintain, if you want, these kind of these maps. So the question is, okay, but what is happening in these regions that lose somehow their specific reactivity to some specific visual information, right? And one idea is that this region will start to tune to non-visual information. So this is one thing that, again, like us and many others around the world have observed, is that if you put a congenitally blind people and you use neuroimaging technique, like for example, functional magnetic resonance imaging, and you'll make them listen to sounds or touch objects. Here I take the example of sound processing, but it's the same thing for tactile information processing. What you see is that these people, in addition to the classical network of auditory regions, for example, the blind people activate the occipital cortex, and massively so, which is not the case in sighted people, right? And if you contrast statistically what is more activated in the brain of a blind person compared to a sighted person when they process sounds, you have this massive activity that is observed in the visual system, right? This occipital posterior region at the back of the brain. So as I've said, like nurture is in the place, right? So this this example that I've just shown you, and there are many others, show you that actually, yeah, the way you experience the world, and for example, if you experience sensory deprivation and you have to mostly rely on the sense of touch or auditions, the regions that are typically processing visual input, they reorganize themselves and process a non-visual modality. It's a phenomenon that has often been called cross-modal plasticity, right? But the point is that what I will try to demonstrate in the rest of my talk is that this cross-modal plasticity is kind of a Trojan horse for understanding brain development. So obviously the obvious feature of it is that experience or nurture shape brain development, right? Because we see this brain reorganization in blind or deaf people. I will speak about that a bit later. But what I'm trying to demonstrate after that is that behind this surface demonstration that there is this cross-modal plasticity, you can find trace of nature. So basically what I'm trying to demonstrate is that the cross-modal plasticity, this brain plasticity, is under specific constraints, so that the expression of this plasticity will be also constrained by a built-in interaction between sensory regions. And what do I mean by that? Is that the way the brain is connected, right, will shape the expression of cross-modal plasticity. So the idea is that you have different networks that are specialized in processing specific kind of information, and that these networks are multisensory. An example I will start with is, for example, audiovisual motion processing. But so here you can extend that to many other type of information processing that are redundant across the senses. And the idea is that there is this functionally specific connection between the senses, and that if you lose one sense, right, this organization will constrain the expression of this cross-modal plasticity. So the invasion of the visual system by sounds will somehow follow some guidance that is that you can find in this multisensory scaffolding of brain regions that I believe is inherited. And this is true for deaf people, as I will try to suggest later on. So let's start with the first empirical results of these talks. So here, like I say, like if you face a computation that you have to do in your environment, like processing motion information, this is a skill that is extremely important for the survival of living things, right? This was true before, right? If you are a predator or a prey, you need to very efficiently process where moving information are coming from or going to, right? And this information typically, and this is true again in modern life, if you want to survive in New York, you have to process very precisely how information moves around you. But these information, they are typically multisensory, right? So they are not only visual. And the classical way to conceive how the brain solves this problem is that you have different kind of energy, they enter different receptors that capture these different energies, and they project in the brain in selective networks, visual networks that will process visual motion, and auditory network that will process auditory motion. And then maybe later on in the brain, there will be some kind of integration. But what we suggest is that these systems have direct link between each other, right? For efficient computation of moving information, you will need actually some sort of quick and direct interaction between these specialized sensory systems to process auditory or visual motion information processing. Right? So therefore, these regions, what we were trying to address here is whether these regions are more multisensory in nature than was previously expected, so that the senses are less separated than what was thought before. And one region that attracted a lot of our attentions lately is a region that has been called HMT plus V5, which is a region in the human brain that is specialized to process coherently moving information. So for example, if you contrast these two stimulations, the one on the top and on the bottom, you will see that you will have in these regions, in the medial temporal, occipito-temporal cortex, more activity when things move coherently versus when they flicker randomly, for example, right? So it's a region that is specialized to process motion information and more precisely process the direction of motion information. You have columns in these regions that are tuned to specific directions of motion. This is known from animal and no human literature. And these columns, they group into hypercolumns, depending on the axis of motion. So left is always next to right, or most often of the case. So it's called an hypercolumn. But what you have to just keep in mind is that inside these regions, there are these clusters or these columns that prefer one specific type of direction, like moving to the left versus moving upward or versus moving downwards, right? So therefore, there is information. We know that there is information about motion directions in these regions. And so what we did with a student of mine, Mohamed Rezk, is to design studies to probe this organization in vision first. So we will present stimuli like those ones, we call them random dot kinematograms that move in different directions to the human brain, and then try to reproduce this sensation of motion with sounds. With sounds, it's a little bit more tricky to do that. So we need to use a trick to reproduce vivid sensation of auditory motion while people are lying in a scanner. So we use this system that we call the squid, basically. It's composed of 64 speakers in this arm that can rotate, and we basically insert in the ear of each participant a small microphones, and we record moving sounds while they are sitting in front of this setup, and we replay this information while they are in the scanner, so that we have what we call age-related transfer functions that are idiosyncratic for each individual. So basically, if you want to have this vivid sensation of motion, especially in the vertical axis, you need these cues. So you need to use this trick. Okay, it's just to a little bit methodological here, but basically using this trick, we can now reproduce, as for vision, the sensation of moving sounds in these different directions while people are lying in a scanner. So the first thing we do is a very simple experiment, what we call a big block design experiment or a localizer experiment. We present moving stuff in any direction versus static stuff in a block design, in vision and in audition, when we record a brain activity using fMRI, right? And when you contrast what is more active when things move versus when things are static, so here it's a contrast to isolate motion selectivity in vision and in audition, what you have is that you have the expected usual suspect for visual motion, which is HMT plus V5 that activate more for visual motion than visual static. You have the planum temporale that activates more for auditory motion versus auditory static. And already you have regions that are interesting, that are less described in the literature, regions just anterior to V5 that prefer auditory motion, and then a zone of overlap, right? A region inside V5 that actually react also to auditory moving information. So regions that process auditory and visual motion information. So now we can localize these regions individually in each subject and do a second experiment and probe inside these individually localized regions for motion direction selectivity. So we do a second experiment. This time we separate the direction of motion in vision and in audition, and this allows us to use, and I don't go too much into the detail, but to use machine learning. So what we call brain decoding, it's some sort of a tool that you use for mind reading if you want, right? To use the pattern of activity link to any directions, right? Either in vision or audition, and you basically use machine learning, right, to try to predict from brain activity what was the direction of the motion that was presented. If you can do that above chance, we say that there is information in the brain that is linked to motion directions. And when we do that in HMT V5, what we see is that if you have 4 directions, right, up, down, left, right, so chance level of a classifier is 25, right, if it was a chance, well, see that at least in the right V5, you can decode significantly above chance the direction of visual, but also auditory motion information. So that is actually very interesting to see that in these regions that as typically, if you open a textbook of neuroscience considered as only a visual region, you can decode actually the direction of motion, not only in vision, but also in audition. And now that we have observed that this region can decode motion direction in both modalities, what we can do is that we can use our machine learning classifier that is here basically a support vector machine to train it in one modality and classify in the other modality, right? Train the classifier on visual data and see if the classifier can learn the structure of motion direction from the visual data and decode auditory data. If we are able to do that, it means that there is an organization in these brain regions that is somehow independent from the modality of the sensory input, right? And what was observed recently in our studies that yes, you can do, right? So in right V5, at least, you have successful cross-modal decoding. So basically you can train the algorithm in vision and decode in audition and reversely in right V5. And therefore this suggests that in these regions, right, there is a processing of motion direction that is at least partially independent from the sensory input. So a truly multisensory or supramodal region, right? In some sort. And when we do a searchlight analysis to see if there are other regions that can do this cross-modal decoding, actually we don't find any other regions. The only regions that can do it is these regions within V5, right? So this is actually pretty selective region in the brain. And we believe that these regions process auditory motion information in addition to visual motion information because there is actually direct structural connection between auditory and visual motion centers. Like I've said before, right, when I do my localizer, right, if I go back to this slide, wait a minute. So here, this slide, you see that there is these regions like V5 that is specialized for visual motion, mostly visual motion, this region that is mostly specialized for auditory motion. And what we believe is that between these two regions, there might be a direct anatomical track that link these two systems to exchange the information about what? About motion processing. And this is what we, a student in my lab that is specialized in a type of analysis that we based on diffusion data. So basically, I'm not entering into the detail of that, but basically able to reconstruct white matter track between brain regions. So basically probing the structural anatomy of the brain. And what we see is that there is this direct connection between auditory and visual motion centers that probably, what we hypothesize at least is that this track allows the rapid, fast exchange of auditory and visual motion information for multisensory integration, right? And so if I go back to my model, somehow very, very schematic model is that what we have seen, at least in V5, is that there is this exchange of information, and that in the visual regions, actually these regions are not only visual, but they also process auditory motion information. So does that link to what I've just said at the start of my talk, or does that link to the blind brain and the constraint that the multisensory constraint? But well, the hypothesis is that if you remove visual input, right, and you don't serve to these visual motion regions, you don't provide visual input because you are born blind, there will be a preferential invasion of this region by auditory motion information because there is this link between these different systems. And I believe that this link is somehow inborn in the human brain. So let's test it. So this is a study that was done by Ceren Batal, actually from Turkey, that did a PhD in my lab. And And she just did a very similar experiment as the one as I just mentioned, but obviously only presenting sounds here because we were also scanning congenitally blind people. And yeah, very similar experiment as before. And the first thing we can do is a univariate analysis. I threshold this map very low. What we see is the regions that prefer auditory motion versus auditory static. You have a network of regions with hotspots in the planum temporale here. And again, we found back, it's an independent study, it's nice, these regions that overlap with V5, right? This more posterior region that is selective to auditory motion. We call this region, we have termed this region MTA, Middle-Temporal auditory or anterior region, anterior from V5. And now this is the brain of sighted people processing auditory information. Pay attention to the slide when I switch. Now this is how the brain network of blind people processing auditory motion information. And what you see is that there is an extension of the network toward the posterior cortex with somehow kind of an invasion, right, of the region that are adjacent to V5, right? If I go back to the network that you see in sighted people with this island here that is also selective to auditory motion, if we look at what's happening in blind people, boom, there's a massive extension of this network. And it gives you a sense that this plasticity, this extension is not a stochastic process. It's not going everywhere. It's just mostly an extension of something that is somehow already there in sighted people. If we contrast the two groups and ask what is common between the two groups, it's all the blue regions. So all the auditory cortex obviously react in sighted and in blind subjects, right? And you have these regions also that are common in sighted and in blind subject that overlap. The white outline here is visually defined V5. And you have these red regions which show the extension of this network in congenitally blind people, right? In what is known to be V5. And now if we go again with a decoding technique where we decode the direction of auditory motion in these regions, what we saw was actually pretty interesting. You can focus mostly on the right side. It's even clearer. It's the same pattern on the left hemisphere, but it's clearer on the right side. What we see is that you have extended auditory decoding in early blind in blue, in V5, so in MT, right, compared to sighted subject. But in the planum temporale, the regions that typically does the job of processing auditory motion, it's still there in both groups, but it's reduced in the blind group. So you have a very nice interaction. It's actually also true in the left hemisphere, where you have more decoding of auditory motion information in what is typically the visual regions, right? And reduced decoding in the temporal cortex, right? As if there was a game of balance between brain regions, where if you observe this cross-modal plasticity, you observe this extension of the networks that are selective to auditory motion information processing, it somehow relieves the computational load in regions that typically do the job, like the temporal cortex, right? So I just showed, I was speaking mostly about blindness, but would that also work with deaf people, right? So basically here, the idea is the reverse. So instead of depriving vision, we will deprive audition, right, in congenitally deaf people. And if the model or the hypothesis confirms, we will have preferential invasion of the regions that prefer auditory motion by visual motion in congenitally deaf people, right? And let's just test that. It's an experiment. We have different type of stimuli here. It was done in a kind of different fashion. And here we now have three groups. We have congenitally deaf people, hearing people, and also a group of hearing signers that are there to control that the effect we see in these brains are not due to the use of sign language, right? Because congenitally deaf people typically use also sign language to communicate, and this also trigger brain reorganization. So we wanted to tear apart what is linked to deafness and what is linked to the use of sign language. So this is why we have this third group. And just in a nutshell, now I will not really present the data of this group because we have not seen nothing very particular in our task with the hearing signers. So hearing signers were mostly similar to hearing controls. and different from congenitally deaf subjects. And so we presented a stimuli like the one you just saw, moving information. People had to do a task like saying if it was moving faster in the left or the right side of the screen, and different type of motion, as you have probably seen, either like horizontal motion, approaching, receding, or stochastic motion as implemented. So let's look in the brain, what is happening here for visual motion processing, but no surprise for you, you recognize these regions, V5, right? This lights up for visual motion processing in hearing people, and this also lights up in deaf people, right? So V5 is lighting it. But now something interesting in addition is happening. There is also a bit of parietal cortex in both groups, but here you start to see in deaf people a region in the temporal cortex that is present and activated in congenital deaf people that you cannot see in hearing people. And if you contrast statistically these two groups, you have one specific region in the temporal cortex that is more activated in deaf people compared to hearing people for visual motion information, right? And where is this region? And it's outlined here with the dashed line here. This overlap with what is known of the regions in the planum temporale that prefers auditory motion information. So again, like we have seen with the blind people, it seems that there is an enhancement of the recruitment of the temporal cortex for visual motion here in deaf people, but not a random extension. It's the regions that typically prefer auditory motion that now react to visual motion in deaf people. And when we do some connectivity analysis, we see that actually in deaf people, there is enhanced, here it's not structural connectivity, it's functional connectivity, but there is enhanced connectivity between the regions that are reorganized in deaf people to process visual motion information and the regions that typically process visual motion information, here V5. And now we do a decoding experiment. So we just, instead of just doing what we call univariate analysis, so what is more activated in each voxels, we now take a group of voxels and decode the direction of motion, as I've explained before. And what we see is that in the temporal cortex, in the planum temporale, you have enhanced decoding of visual motion in early deaf compared to control subjects, right? So here, hearing subjects, signers or non-signers, that show similar activity. And again here, the same pattern, but reversed as the one we have observed in blind people, is that you have a reduction in the decoding of visual motion in V5, right? So the region that does the job, you see that the decoding accuracy are skyrocketing here, are really, really high compared to chance. And what we see is that here, you can decode in every group, right? But still here now the decoding accuracy is higher for the hearing people compared to the deaf. And again, you have an interaction between these regions, right? With more decoding accuracy in the temporal cortex of the deaf and less decoding accuracy in the occipital cortex of the deaf people, right? And again, suggesting that there is this gain of balance. Like if this cross-modal plasticity that is selectively recruiting the regions that are typically auditory motion selective, release the computational load of the regions that typically do the job, which is in vision V5, as we have seen before, right? So I just like look how I'm going with the time. So I think maybe I still have like 15 minutes. Am I correct?
Yes.
Okay, perfect. So I'll go to somehow like try to demonstrate the same thing, but like switching a little bit gears. As a Belgian person, I did not resist to present these pictures that probably most of you have already experienced or seen. It's a painter. It's called René Magritte. He was actually living 200 meters away from my house now in Brussels, and this person painted these figures, this painting, saying like in French, ce n'est pas une pipe, so this is not a pipe, right? While showing a picture of a pipe. And the idea about that is obviously this is not a pipe because the pipe is much more than the image of it, right? So with the pipe, you have the real pipe, you can touch it, you can smell it, you can somehow hear it when you use this pipe and see it at the same time. So the concept of a pipe is much more than the image of it, right? So this is actually the fallacy of the image, as he calls it, right? And so basically, why I'm interested by that is that because we have in the occipital cortex also regions that prefer processing objects, right? Some specific type of objects. So you have regions that prefer processing manipulable objects, regions that prefer processing faces, for example, the fusiform face area, and other regions that prefer processing places or buildings, right? So it's 3 paintings from Magritte here that I used to represent this category selectivity that you can find in the occipital cortex of sighted people, right? But as you can guess now, our model is that maybe you have the same thing in the auditory domain, right? Where you have auditory categories that prefer recruiting some specific temporal regions. And what we believe is that there is an interaction, that these modalities are intertwined, and that somehow you will observe in these regions that are thought to be purely visual, also activity for sounds processing following this categorical organization I was just mentioning. And that if you deprive the system from vision or audition, you will have this preferential extension of sounds following this categorical organization. So this is a study that Stefania did in my lab and published last year in eLife, if you're interested. And so we scanned here groups of subjects, groups that were doing the experiment in vision, sighted people, blind people doing the auditory version of the experiment, and a third group of sighted people doing also the auditory version of these experiments. We presented different types of categories in vision and in audition using such type of event-related design where we were scanning, where we were presenting basically sounds of birds, sounds of humans, like a voice, basically sounds of animals and these things. And we were presenting that in vision and in sighted subject, and in audition in sighted and blind subjects. So first of thing we did is what we call a winner-take-all approach about these 4 categories that we presented, and to extract which region in the occipital cortex prefer each categories. And as I have described, what we see is that there are some regions that prefer some categories, like the more medial regions here, overlapping with the para-hippocampal cortex prefer places, right? The seeing places or buildings. And you have just next to it regions that prefer seeing faces, around that regions that prefer seeing animals, and then even more lateral regions that prefer manipulable objects, objects that you can manipulate, like the pipe, I was saying. Now let's look what's happening in these regions when you don't present the image of these things, but the sounds of these things, right? And let's first do that in sighted subjects. So obviously the overlap is not perfect, right? But you can find that the structure of the activity is somehow similar, right? You have medial regions here that prefer big objects. Next to it, regions that prefer here voices, like human sounds, and a little bit more lateral here, regions that prefer manipulable objects, for example. Right? And if actually you correlate these 2 maps together, you see that there is a significant correlation between the organization of this region. So this is already pretty interesting to observe that even in sighted subjects that process sounds, they recruit actually these visual regions in following an organization that we know is the organization present when these people process visual information. But now maybe some people may say, okay, this is visual imagery. Sighted people, they hear these sounds, they create the image of these objects in their mind, and this is why you have this correlation. Obviously, visual imagery is out of the game when you speak about congenitally blind people that have never seen, right? And what we see is that this organization, we can also find it in congenitally blind subjects. If anything, the correlation is even stronger between the congenitally blind subject doing the auditory version of the task and the sighted subject doing the task in vision, right? So similar region seems to prefer similar type of categorical information across vision and audition in both sighted and blind subjects. So now we can do a little bit more fancy analysis. I not go too much into the detail, but you can take each of the categories for the simplicity. Before I was just speaking about 4 categories. just to make these maps more understandable, but actually we presented 8 categories. So we can decode pairwise these categories together, and a vector of decoding of how these regions decode all different categories, with the idea that if 2 categories are closer together in the brain, the decoding accuracy will be lower. And if 2 categories are further away in the way they are represented in the brain, the decoding accuracy will get higher, right? So this is this peak and valleys that you see here, this chain of mountains of brain decoding activity here, just reflect the way the occipital, ventral occipito-temporal cortex organize the distances between information processing in the brain. And you can basically represent, so you can decode this information in the early visual cortex, primary visual cortex, or these more higher level regions. No surprise, you can do the category decoding. Since it's pairwise, the chance level is at 50. You see massive decoding accuracy in vision in these 2 regions as expected, right? We are in the visual cortex. But already what is interesting is that when you look at blind people in orange and sighted people in blue, in the early visual cortex, you can decode auditory categories in blind people, and not really much in the sighted people, but in VOTC, you can do those groups. Even though it's also higher in blind people compared to sighted people, it's also there in sighted people, right? You can decode auditory categories in the ventral occipito-temporal cortex of sighted subject, as you can do in blind people, right? And if you look at how this information is organized, so you can create these dissimilarity matrices between the decoding accuracies across different categories and correlate these matrices together. And what you will see is that these matrices again correlates. What does that tell us is that the way the ventral occipito-temporal cortex organize the processing of auditory categories, so these peaks and valleys, like I was, the distance between different categories, is somehow similar in vision in audition. So obviously, the decoding is much higher in vision in sighted subject in the occipital cortex. This is obvious. This is still visual, are still visual regions, but you can still find information while blind and sighted subject process these sounds, and the shape of these peaks and valleys is somehow similar across the senses. So 2 categories that are more distant in vision are also more distant in audition, right? So this is to us very, very interesting. And now we did what we call representational similarity analysis, contrasting our brain activity with some models, but I have the feeling I'm saying a lot here, and I kind of skipped that, but in a glimpse, what you just have to remember again is that what is encoded in this region, in ventral occipito-temporal regions, is somehow high-level categorical information that is independent of low-level features in vision and in audition. So basically, the concept of an object being a face, a voice, a manipulable object, this is what seems to be encoded in blind and sighted subjects in these regions, and not the low-level features. And just to take an example, and I'll finish on this last example before concluding, here, just because I understand what I've shown is pretty complex, but this is maybe more understandable. Here, I've highlighted in orange the regions that prefer faces in sighted subject, right? Regions that prefer faces than any other type of pictures I present to them, right? And in yellow here, it's the regions that prefer voices in sighted subject, or in blue here, voices in blind subject. And you see this very, very nice overlap between the regions that prefer faces and the regions that prefer voices, right? Compared to any other type of information or categories that I've presented, right? So it seems that there is this correspondence. And obviously this correspondence to us is not random. It's obviously because we systematically face and voice together. We integrate this information to extract speech, gender, age, trustworthiness, whatever, name it. There are many information that we extract simultaneously from face and voices. And so therefore we believe that there is this exchange of information between these regions, as I've demonstrated here. And so if we do the same thing with the deaf, now you know my trick. I test my model in deaf people also. And I present faces and houses in this case. And what I see is that there is one region in deaf people compared to hearing signer or hearing controls that prefer faces compared to houses in deaf subjects in the temporal cortex. And now you can, I guess, guess where is this region? It's actually typically the region that prefer voices in hearing people, right? Again, like the region that reorganize in the temporal cortex of deaf people and enhance its response to faces is actually the regions that originally is liking voices in the hearing brain. And actually, the more the deaf people recruit these regions, the better, sorry about that, the better they are in processing faces, right? As assisted behaviorally. We did a certain experiment just to show that these reorganized regions, it's called typically the temporal voice area, discovered by Pascal Belin. Now we call this region the temporal face area in deaf people, right? And these regions not only react more to faces than houses, but also react more to when we present different faces than the same faces. So basically that these regions show adaptation, right, or release from adaptation here as probed by this typical design of presenting same versus different faces. And so basically it tells us that these regions not only react to face compared to us, but also can recognize different faces, right? So somehow is engaged in face identity processing. And then we did an MEG experiment, magnetoencephalography here to have the time course of this activity. I'll go very quickly here, but to show that the activity in this reorganized region processing faces in the temporal cortex of deaf people, if anything, reacts very quickly after the fusiform face area, the regions that prefer faces over houses in the occipital cortex in deaf people. And again, we believe this is because there is this direct track, anatomical track, linking the fusiform face area and the temporal voice area. Actually, this was discovered by a German group the name of the PI is Katharina von Kriegsten, and we confirmed that using DTI, we confirmed this and extended to deaf people to show that deaf people also have this track, and we believe they use this direct track linking FFA and TVA to send information about faces in this temporal region in the reorganized temporal cortex of deaf people. So it's my penultimate slide. So what I've tried to show you here is that, okay, as we know since 30 years now almost, there is this cross-modal plasticity in sensory deprived individuals. So the deprived cortex react to the the remaining senses somehow, but that this cross-modal plasticity is somehow constrained by the multisensory architecture of the brain. That there is some kind of these regions, they keep somehow their functional role. They change. What we have seen is that the fact that sound processing extend in the occipital cortex of blind subjects release somehow to some extent the computation in the temporal cortex, and reversely in deaf people, the extension of visual processing in the temporal cortex somehow release the computational processing in the occipital cortex, like if there was this gain of large-scale redistribution of computation. So the last slide, and I started with Plato. So when you look at cross-modal plasticity and the expression of cross-modal plasticity in the brain, what obviously is striking is this expression of nurture, right? Experience change brain development. We have regions that were typically considered visual, now they massively respond to sound. Here I mostly used sound, but this is the same thing for touch, right? But actually behind the scene, there is somehow also an influence of nature, right? So basically here, like the cave allegory is that this cross-modal plasticity, actually behind this cross-modal plasticity, there might be an inbuilt multisensory architecture that constrain its expression. Sounds, when they go to the occipital cortex of blind subject, they don't invade this region, they don't hijack this region randomly. specific sounds go to specific places of the occipital cortex in link with the function of these regions. And yeah, so to finish, I'll thank my lab that have done all these very nice work, right? It's hard to do these studies because they are technically challenging, but also you have to recruit these participants, sometimes not easy, so it provides an extra challenge, but I'm very lucky to work with very motivated people, so we can do these studies, and obviously these funding agencies, and I'll finish by thanking you for your attention today. Thank you.
Thank you very much for your insightful presentation. We're gonna give a 5-minute break, and then we're gonna proceed with the Q&A session.
Okay, so I'll be back in 5 minutes then.
All righty. So if anybody has questions, please raise your hand, and I would like to encourage you to share your video cameras and your voice. Also, like, if you're not feeling comfortable of your question being recorded, we can also cut it later on so that once we publish the event, it won't appear there. So yep, so I'm just checking if anybody raised their hand. Mark-Jan, go ahead.
Hello, thank you for your brilliant presentation. I might have missed it, so apologies if you mentioned it already. But I often saw fMRI data, which might lack temporal sensitivity. And I was wondering what you could say about the temporal aspects of your model. And the reason that I'm asking is because it is often observed, especially in the studies of motor learning skills, motor skill learnings, that when imagining a movement, the parts of your brain gets activated as if that movement is indeed initiated. And that can even help you learn the movement a lot easier than just practicing it. So how much of the brain activation across modalities could be accounted by such human capacities? And I was wondering specifically, are those brain activations also temporally matched with the presentation time of the stimuli?
Yeah, thank you for the question. And I think there are two questions in one somehow. So the first part is about the temporal resolution of fMRI, and fMRI is terrible for temporal resolution, right? So you cannot say much about temporal resolution from fMRI. So I don't know, maybe you are familiar with that, and what I say is trivial, but so we acquire slice, I mean, an entire brain volume somehow every typically 2 seconds, let's say, right? So, which is in terms of brain processing is a world, right? So literally it's not a technique that is used to really inform anything about time. So that has to be said, and this is due to the fact that it relies on somehow vascular changes, right? So the what we call the BOLD effect, the blood oxygen level dependent effect. So we basically record a derived product of brain activity, which is the vascular change that is linked to this brain activity, and this is very slow to emerge in the brain. So basically, this is why this technique is not really used to say anything about timing post-processing, but at one point of the talk, and I was very quick on that, and it was at the end because I saw that I was running against time, but I say that I kind of did it's the deaf study that I did where presenting faces, and I did an MEG study, so a magnetoencephalographic study where, as you may know, we have milliseconds precisions about brain activity. And what we saw is that we could see that these regions that process the reorganized input. So let's say in the case of deafness, the visual input, like faces, for example, they react extremely quickly post-stimuli presentation, like less than 200 milliseconds post-stimuli presentation, which is very similar to what you observe in face selective regions, right? Face selective regions like FFA typically reacts 170 milliseconds after presenting a face. So it's the classical N170 for face versus houses, for example. And we saw that it was 190, for example, in the temporal cortex. So this temporal information let us know that it's very quick after that, right? It's not a slow process that is relying on many top-down effects that will come back, which maybe brings us to the second part of your questions about like, could it be some sort of mental imagery that people are doing, like when you do motor imagery, like you said, and these things, right? Which is a very prominent features of how we process information. And I totally agree with that. So inside it, I mean, in non-deprived individuals, this is true that it's always somehow, if you want to confound, right? So I'm here presenting moving sounds, right? And I have activity in regions that typically process visual motion information. How do I know that it's not just that people see in their mind's eye, right, things moving, so that they imagine visually things moving, and therefore, because they imagine it visually, they would activate visual regions, because we know that imagery, like motor imagery, activates motor cortex, visual imagery activates visual cortex, and auditory imagery activates auditory cortex. So how do we know that? We are never sure about it. It's always a possibility, right? But one strong information that suggests to us that it might not only be that is because congenitally blind people also have it, right? So congenitally blind people, they cannot do visual imagery, right? They just have no visual representation. They never had visual experience. Like a deaf person cannot do auditory imagery, right? They don't have an idea of what is the sound of a voice, for example, or blind people do not have a visual representation. They could obviously create some kind of tactile representation of these things, but at least it is probably something that is behind the vision. Then we do a little bit of more technical trick to try to probe these questions. I don't know, I mean, maybe I go too much into the details, but typically what we see in the studies that do visual imagery is that when you do visual imagery, you also activate your primary visual cortex, for example, right? So we saw that if these sounds activate these regions because they do visual imagery, we would also see activity in primary visual cortex, which we don't typically. So all this together, the most parsimonious explanation for us is that it's not imagery, but at least in sighted people or hearing people, I cannot guarantee 100% it's not playing a role.
Great. Yeah, thank you. Thanks so much.
Thank you for the question. That's a great question.
All right. Ahmed, please go ahead.
Hi, thanks for the talk. So early on in your talk, you showed that B5 also activates to auditory motion in sighted individuals. So then in light of that, why would you continue to call B5 a visual region? Why not call it a semantic region that kind of specializes on semantic category in this case? Emotion. So in which case, what you're talking about is not cross-modal plasticity so much as reorganization of the semantic system.
It's really an excellent question, again, and a question I like very much. So if you allow me, I can go back to my slide and I'll show you something. Do you see my slides?
Yeah.
I have it somewhere, I'm sure. Let me here. So I don't know if you are familiar with these fMRI and stuff or not really. I mean,
yeah.
So okay, basically here your question is interesting. So why do we still call them visual regions? So V5 is, the V of V5 is for visual region 5, right? So why do we still keep to say it's V5? It is actually because these regions, what I'm saying is that there is auditory information in this region, and that this auditory information follows an organization that is the one that we observe also in vision. So basically directional selectivity, right? It's not only that this region activates for moving sounds, it's that also it activates similarly, partially similarly for the direction of motion. But when you do a multidimensional scaling, so basically you try to take the activity of V5 when it reacts to sounds and vision and plot it in 2 dimensions. It's some sort of independent component analysis, if you want, but call it multidimensional scaling. What you see is that the first thing the algorithm will do, right, in the first dimension is to segregate visual and auditory information, right? It will plot, here you have the 4 directions of visual motion, up, down, left, right, and in audition, up, down, left, right. And the first thing the algorithm will do is that it will very easily segregate the auditory and the visual information. But on top of that, what you see is that inside these segregated clusters in virtual space here, the dimensional space, is that there is an organization that is similar, which is what we call an axis of motion organization. Up goes with down and left goes with right, right? And this is true in vision, in audition, right? And this is what we believe is linked with the neurophysiological structure with this column and hypercolumn I've mentioned at the start of my talk. So basically what we believe is that the ability to do this cross-modal decoding is linked to the fact that the way things are represented, so this axis of motion, is respected between the 2 modalities. But as we see, and this is really striking, it does not mean that the global activity in this region is identical between vision and audition. Otherwise, these clusters, they will overlap in these things. And now to go a little bit beyond this illustration here, is that, and this is a study we are trying to do now, is that I'm not sure that if you will do a lesion of V5, right? Imagine now you do a lesion of V5. For example, you will use transcranial magnetic stimulation, you create this transient visual lesion of V5. You will, as we know, disrupt visual motion information processing. I'm not even sure we will disrupt auditory motion information processing, right? Even though I'm observing information there, right? This is something I'm trying to observe now. So how does that work? Still, you have the planum temporale, right? This region still is the hotspot of auditory motion information processing. And what I believe is that these regions project information to V5 in a co-registered fashion, axis of motion, most likely for multisensory integration, most likely for situations where you have both at the same time and you need to co-register these 2 things, right? But I'm not even sure at the moment that this information, that auditory information that I observe in V5 is mandatory to process auditory motion. You see what I mean? This is a little bit subtle, but this is why I would not dare to call this region a semantic region, right? Or a supramodal region. I've argued with some colleagues about that. But I still believe we are not entitled to call these regions amodal, semantic, whatever, because of what I've said. I think these regions in sighted people still is dominantly a visual regions, even though it has also co-registered auditory information, most likely for multisensory integration.
Because in sighted people, most of the time motion is processed in visual modality.
I did not get the start of your question.
Sorry, I said that could it be that the reason be that in sighted people the semantics of motion primarily develops via visual modality?
Yeah, this is a good point also. This is a good point. There is surely a visual dominance for processing motion information. And we see that when we, for example, present incongruent information in vision and audition. So let's say something that move to the right in vision and to the left in audition, what people tend to do is to respond right. So the visual modality follows the visual modality because there is this sensory dominance for that. And so this may be why also these regions attract somehow, if you want, auditory input to create this representation. I mean, when I say it's why I don't dare to call this region a semantic region, I mean, this is debatable. This is really a hot topic. Some people have actually presented words like left and right and try to see if these words elicit left and right activity in the occipital cortex. And there they had like very mixed results. I think nothing really emerged clearly, at least in V5. Some have claimed you could find linguistic information about motion direction, and others have said, no, I've never seen that in my data. So here I would not dare to say that it's a robust result, but clearly people are chasing after it, yes. Yeah.
All right. So Marc-Jean, do you have any other questions? I see that you haven't lowered your hand.
I'm sorry, that was a mistake, yeah.
All right, that's fine. Let's see. Is there anybody else that wants to ask a question? If not, I will do. All righty. So I was gonna ask about, so, you know, a part of your, like, the title of your talk was related to, you know, brain development. So I was wondering, like, if, how would your research would apply to, like, the development, like a children's brain development, and, like, you know, blind children or like deaf children, and like how the connections within their brain would differ compared to an adult.
Yeah, this is also a very, very good question. Thank you very much about that. So one thing, a model that has always been used somehow to refer to how this cross-modal plasticity links to some periods in brain development is obviously, there are two things: the link of with the sensitive critical period stuff, right? So where the plasticity of the brain is the highest. So one thing I can say is that surely enough, if the visual deprivation is very early on, so from day one, so congenital blindness, you have more plasticity than in people that acquire visual deprivation later on, right? So on the top of a developed visual system. You still have plasticity in late blind, but this plasticity is less. So that is a little bit in link with the sensitive periods of brain development. Then the second thing that has potentially more in link with your question is that, as you may know, there is a phase of overproduction of synapses at the early phase of brain development, and then what we call synaptic pruning, right? So basically the jokes that we make about that is that a 1-year-old baby has twice the synaptic connection as the pediatricians that follows him, right? So and this is practically true, right? So you have this massive, overly densely connected brain that is pruned, and these connections are pruned away by experience over many years after that, right? So you have a peak at 1 year old, and then it decreases until adolescence, and you have this pruning that's happening, right? And so what we believe is that this densely overconnected net of connections also include intermodal connections. And this has actually been demonstrated in animal work, where you have actually more connections between, let's say, the temporal and the occipital cortex in a young kitten, right, for example, than in adult kitten, right? So as if these cross-modal connections also are pruned away, like my other question, connections. And what we believe is that in the absence of competitive visual input, in the case of blindness, for example, some of these connections are not pruned away, right? They are maintained in these brains. and explain this potentially this extended auditory activity that we massively observe in these regions. So I believe that in a developmental term, yes, there is probably connections that disappears in typically sensory developing people that are maintained in deprived individuals. I would really love to do developmental studies with blind or deaf babies, right? They are obviously very challenging to do, right? So for very practical reason, scanning normal babies is already a challenge, right? Scanning sensory deprived babies is an extra challenge. And probably what we would like to do, if I were to do these studies, is to stimulate them with sounds or vision, right? And these things, which is also an extra challenge because there are several studies that are done with babies where you just do resting states or DTI, right? So the baby sleeps basically in the scanner, right? Actually, you make the baby sleep. When he sleeps very deeply, hop, you put it in the scanner and then you record his brain activity. But I'm not very much interested in that, I have to say, right? Because I would like very much to stimulate the brain of these babies with sounds, for example. And that is possible, but very challenging. So now, even though I'm very fond of fMRI, I'm also developing electrophysiological studies like EEG studies, and probably I'm moving a little bit more toward this direction in the future because it seems to me just logistically much easier for the type of stuff to do. I will not be able to have the same inferences about spatial localization very precisely as I'm doing with my fMRI. I will have to lose that, but I will gain other stuff on the side, and I'm sure it might be potentially interesting, but yeah, great idea.
Thank you so much for the answer. And I actually had one more question. So I think that like when we're talking about like sensory, sensory information, multisensory information, I believe like other sensory information like olfactory information is understudied compared to, vision and auditory perception. So I was wondering like, how would your research maybe a new line of research that based off of your research on olfactory perception would like, what kind of like conclusions would we end up with that? Like, could you foresee like doing another line of research based on that? Because I would, if I remember correctly, the olfactory cortex is also in like around inferior temporal areas, so that it might be like, related to it, but I don't know, like I have to wrap it in my mind.
No, it's a good point. Actually, I've worked a little bit in olfactions with some colleagues when I was postdoc in Montreal. Had the chance to collaborate with Johannes Frasnelli or Johan Lundström, that are researchers actively involved in understanding olfactory processing in the brain. And so I was involved in some studies. We have actually done some olfactory studies with blind people. I'm revising at the moment, really literally last week, a neuroimaging paper on olfactory processing in blind people. So this is a timely question. I'm a collaborator on these studies. I'm not the main investigator, I have to say. But yeah, this is definitely interesting, right? The point I want to make is that, and you started with that, is that the olfactory system, how the brain, human brain process olfactory information is much less known than any other senses. Vision is for sure the most overly investigated sense, then comes the others, and the olfactory is probably at the end of the line, right? Even though it's extremely interesting, and with COVID now we see this emergence of studies, because one, obviously, as you know, one of the things that is linked with the symptomatology of COVID is anosmia, or partial anosmia, or parosmia, so the experiencing others that are not out there. that actually have done the experience myself because I had COVID. And this like also re-raised somehow my interest in the topic personally. And I'm doing now also some studies. And this is really the early phase, so I don't dare to speak much about that. We have no results, but with anosmic people. So I'm very much like, as you have understood from my talk, I believe sensory deprived individuals are an excellent models to investigate brain developments and what is plasticity, right? And reorganization, and how this plasticity is in link with an inbuilt system in the brain and these things. So I'm a strong advocate of the sensory deprived brain, I would say. And so therefore, in this context, I'm also interested in how anosmic people actually process information with potentially the same type of stuff we have seen before, like this invasion of the olfactory regions by other senses. It's a little bit more tricky, as I said, because we don't know much about how this information is naturally processed in these regions. So we probably have to start with just understanding a bit better these regions before moving on to anosmic. But just like in a nutshell, what quick study we just run with a master student in my lab, she's defending a master thesis now, is how anosmic people think about others stuff, right? So for example, how do they think about food or food-related items or items in our world that have a very strong olfactory components, right? So they're part of our conceptual systems. So I cannot think about a rose without thinking about the odor of a rose. I cannot think about whatever my preferred dish without probably the olfactory stimulation of this dish. But how do these anosmic people think about these things? They have a conceptual representation of these things that miss the olfactory part. Does that mean that their concepts are different? I mean, this is the type of questions that I find interesting.
Yeah, actually, I was literally thinking about that, like when you were talking about, visual motion perception and like auditory motion perception. Maybe it's because it's lunchtime in here, but I was thinking about like how like olfactory perception would be applied to it. Like, let's say the food was just like moving on the table to the left and right and somewhere else, like how, because it's definitely going to interact with the visual cortex because they're also seeing it, but the olfactory cortex as well. So I was just like wondering about like what kind of interesting results would apply to it. Yeah.
Yeah, definitely. It actually, what you are saying is that, and in link to what I say is that, yeah, the senses that are intertwined, right? You always process, you are living in a multisensory world with different sensory organs that capture all these kind of energies, and the brain needs to integrate or segregate these things, right? So obviously you have brain networks involved in this process.
Thank you so much. So is there anybody else left who would like to ask questions?
They all went to lunch because it's lunchtime. So and I understand them. it's dinner time here. So
yeah, it's dinner time there, lunchtime here. All right. I would assume that was all the questions that we had. Thank you so much for being with us today. And I personally truly enjoyed your talk, and I can see in the comments and in the chat that like everyone else also enjoyed your talk. Thank you so much.
Thank you very much for the invitation. It was my pleasure. Bye-bye.
Bye. Have a great rest of your night.
Thank you. Bye-bye.